The name of media agar. Na. What is nutrient agar?
Is
It's used to culture.
It's a basic media.
base.
Used to culture or to non-fastidious bacteria, such as E. coli, Staph aureus, okay? Basic media. Support the growth of non-fastidious, what is non-fastidious in
Which
Farsi?
can't grow everywhere.
In Farsi.
Which do not have
Non-fastidious in
which do not
Farsi.
have very specific nutrient requirements.
In first,
غیر سخت.
What is done?
غیر
can't go into
Saffron.
سخت and fastidious.
Not,
Fastidious سخت
yes.
Okay. Plate contain two sections, two parts. Back of the plate, lid or cover of the plate. For labeling, we use the back of the plate. In the labeling, you must write your name. What is your name?
Bijan.
Bijan.
Yes.
Yes. Here, Bijan. Your lab ID.
48.
Okay. And the name of medium. What is the name of medium? Nutrient agar. For labeling, use the edge of the back of the plate in this area, no center of the plate. Okay? Bismillah.
Okay.
Take CD marker and label. Okay? Solid media in plate, solid media in tube, in the form of a slant medium. A slant in Farsi sheetdar. Pay attention. A slant area and bottom area of the medium. And liquid media. Liquid media without agar. Solid media contain agar. And semi-solid media such as SIN, sulfide, indole, and motility is the name of medium. SIN. And according to the function of media, there are 4 groups of media. Basic media such as nutrient agar, nutrient browse, used to culture of non-fastidious bacteria. For example, E. coli, Staph aureus. Basic media plus growth factor, for example, serum, amino acids, blood, change your media into enriched media. Enriched media such as chocolate agar, blood agar. Enriched media used to culture of fastidious bacteria. For example, Streptococcus pneumoniae, Neisseria meningitidis. These bacteria are fastidious bacteria. Need enriched media for growing. Third group of media is selective media. Selective media such as mannitol salt agar. Mannitol salt. This medium contain salt. Salt inhibit the growth of bacteria that's sensitive to salt and allow to grow all other bacteria. For example, Staphylococcus can grow on this medium. So this medium is selected for Staphylococcus. All Staphylococcus can grow on this medium. It is selected for Staphylococcus. Fourth group of culture media is differential media, such as mannitol salt agar. This medium contain mannitol. I told you all Staphylococcus can grow on this medium, but only Staph aureus ferment mannitol and change color of medium from pink to
Golden.
yellow. So according to this reaction, you can identify a Staph aureus from coagulase negative Staphylococcus. So it's differential media or indicator media. You understand? Mm-hmm. Yes. Finish your labeling. All?
No.
Finish your labeling.
No.
You write your plate. Inoculating tools. Glue. What is this? And needle. Very good. Glue and needle. Needle and glue. You must sterilize your loop and needle before and after of culture. You understand? Yes. By flaming. Take your loop and needle and hold in your hand and hold in top of the plate until to change the color of needle.
Needle and loop.
Can you see it, Mr.?
Yes, yeah, I can see.
Yes, if photomolators we can see.
After that, you must wait for air cooling, 10 seconds.
Okay.
Because heating kill off your bacteria. Don't wait it for cooling of your plate. Wait 10 seconds for cooling. Yes?
Yes.
Okay. Culturing of plate. There are 3 types of culturing on the plate. Liquid medium and
Some.
a slab medium. To culture on plate, divide your plate into 4 sections. Give me CD marker. CD marker. A, B, C, and D. Yes? Place your plate near the flame. Open plate contain bacteria. This plate contain bacteria. According to your lab ID, for example, number 1, number 2, number 3, you must choose your unknown bacteria. First, sterilize your loop. After that, near the flame, open your plate. Plate that contains bacteria. Look at here. This is bacteria. Yes? Can you see?
Yeah.
Bacteria. Yes? Then touch your loop on surface of agar. Don't push it. Touch it. And pick up bacterial colonies. A loop is enough. Okay? Then place your loop in center of part A. Part A. And spread out in all area of part A. Yes? Yes? Part A. Clear?
Yes.
After that, sterilize your loop.
Again.
Then sterilize 5 lines from part A to B. 5 lines. Punch cut. 5 lines. Okay, like this. A to B. 1, 2, 3, 4, 5. You understand?
Why?
Because
1, 2, 3, 4, 5. Yes? Then repeat your sterilization of loop. And repeat again. Sterilize 5 lines from B to C.
take another material.
Clear? Repeat again. and 3 to 4 lines from part C to D.
PUC.
Pay attention.
خمسة.
There is no overlap
خمسة.
between
خمسة.
part D and A. Part D and A. The name of culture is isolation method or strict pure culture method. In this method, the number of or the concentration of bacteria decrease from part A to D. And you can see single colonies of bacteria in part C and D. This method used to isolate out bacteria from mixed culture, from clinical samples. Maybe clinical samples contain 2 or more than a bacteria. By this method, we can separate different of bacteria. Each bacteria contains special colonies according to the size and color of colonies. Pay attention to your plate. What is the color of this bacteria?
White?
Yellow.
Yeah.
And this is white. So some bacteria show white colonies, some show pink colonies, some show lemon colonies, and some show red colonies. For example, red colonies for Serratia marcescens. Serratia marcescens contain red colonies. Do you understand?
Yes.
Culture your plate according to isolation method. A, B, C, D. A, B, C, D. To culture, to culture on a slant medium, we use fish-tail method. Fish-tail method. For this, you must use needle, no loop. Needle. Insert your needle. Touch your needle in the surface of bacterial colonies. Take very small amount of bacteria and pay attention. Near the flame, open your tube. Hold in your hand the cap of the tube. Don't place on the bench. Steril the neck of the tube like this. And then, first, insert your needle in the bottom of a slant medium, like this. Yes? Clear? Then, in a slant area, culture similar as zigzag. Okay? The name of method is fish-tail method. Fish-tail method for a slant. To culture in liquid media, this medium, you can use loop or needle, both. Take tube, remove tissue or cap of the tube, okay? Steril the neck of the tube. Then, near the flame, tilt your tube. And semi-solid. Look at here, this medium is SIM, semi-solid media, SIM. M from motility. We distinguish the ability of microorganism, motility on this medium, SIM.
We should use another.
Remove tissue, don't place on the bench, pay attention here, hold in your hand. Tissue and cap. Steril the neck of the tube, tilt it, and insert bacteria in the and mix it. You understand? No, this one.
Do zigzag.
Yes.
Like this.
You understand? Yes. That's right. And place on the rack. This is a rack for tubes. And still your low pour meter. Pay attention to transfer bacteria from liquid media to solid. We use loop. Liquid to solid. We use loop. To transfer from solid media to liquid, you can use loop or needle, both. You understand? Yes. Let's spell that. Slant. Slant in Farsi is sheep. Is slant. Okay? Sheepdog. Sheep. Farsi. لهجه فارسی خوبی داره. Slant medium. The name of medium is mannitol salt agar. MSA. Mannitol salt agar. This medium is liquid medium or browse medium. BROTH. Browse medium or liquid medium. Without agar. This contain agar. I think is 1% of agar. Solid medium, this is in the plate, contain 2% of agar, 2 and more percent of agar. Okay? And this medium is Simon citrate, used to identify different species of enterobacteria.
Simon?
Which Simon citrate or citrate utilization test. In this medium, we check the ability of microorganisms to use of citrate as a sole source of carbohydrate. Bacteria that use citrate change the color of medium from, what is the color of medium? Green. Change it to blue. Green to blue. Blue is positive reaction on this medium, meaning that bacteria use citrate. pH indicator in this medium is bromothymol blue. Bromothymol blue. To culture on solid medium, there are different methods to culture in solid medium. Method, isolation method, or a strict pure culture method. For this, give me look.
Loop and needle.
Barman.
Doctor.
First, aspirate your loop.
I will leave this to you.
Yes. You can distinguish staphylos from other staphylos. To observe bacteria, we use staining. There are two basics of staining here. Simple staining and differential staining.
شماره شما چنده؟ چون شما دوتاتون نوشتین 12. اون شماره نیستش که شماره که روی لیست نوشته. این هم شماره که
and differential staining. Okay. Can you explain about the basic shape of bacteria?
The basic shape is
Bacillus, oxide, or spherical, bacillus or hood shape, and spherical. Bacillus or hood shape in Farsi is milane. You understand? Milane. Coccus or coxide is spherical, yeah, the same, yeah, okay? And a spiral is martichi. A spiral bacteria is spiral.
نه گفت من بریش شدم
Syphilis, okay? And root-shaped bacteria and coccus. Okay? Milei, yer, marpici. Root-shaped or bacillus, bacillus plural B. Coccus or cocci and spiral. Okay? And different arrangement of bacteria. For example, Diplococcus. This is Neisseria. Gram-negative Diplococcus. And pneumococci, this pattern. Gram-positive Diplococci. Gram-positive Diplococci. Gram-negative. Tell me the example of gram-negative Diplococci. Neisseria meningitis. Neisseria gonorrhoeae.
دکتر من معذرت میخوام ولی درست شده ها.
درست شد؟
بله.
مرسی. In differential staining, we use two or more stain. For example, in gram staining, we use crystal violet and saffron. Crystal violet and saffron. And after staining, you can distinguish bacteria according to their reactions to staining method. Gram positive from gram negative. Gram positive color is
Violet.
purple. Gram negative
Red.
is
Saffron.
pink.
Pink.
Come here.
حاسبها.
Is that pink? Has a
Right.
Red. Okay. In simple staining, after staining, you can see shape, size, and the arrangement of bacteria. Shape, size, and arrangement. In differential staining, for example, in gram staining, in addition to shape, size, and the arrangement, we can distinguish gram positive from gram negative. Mr. Dai, what is the color of gram positive?
Yellow.
Yellow.
Positive? Violet.
You want to stay in class? Pay attention. No, no. Color of gram positive bacteria?
Purple.
Purple. Show the color of which stain? Gram positive.
What?
Crystal. Violet. Gram negative or pink. Show the color of saffron. Okay? Explain about the differences between gram positive and gram negative.
يلا روح يلا.
You.
Purple and pink. The gram positive is purple and the gram negative is pink. It's and the pink one is used. What was this one? This first.
No. Explain about the structure of bacterial cell wall in gram negative and positive.
The gram positive have a wide peptidoglycan and don't have the outer membrane, and the gram negative have outer membrane and inner membrane.
What is LPS?
LPS is
Lipopolysaccharide or endotoxin. 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 We can see which bacteria? Gram positive. No. Gram negative bacteria contain outer membranes.
Yes.
Okay, outer membrane is special structure in gram negative bacteria. In gram staining, we remove outer membrane by alcohol. You understand? Gram-positive and gram-negative bacteria contain peptidoglycan. Crystal violet react with peptidoglycan. The peptidoglycan layer in gram-positive is very high. And so, crystal violet, because crystal violet react with peptidoglycan, you can see purple after staining. But outer membrane inhibit the penetration of crystal violet. So we need second stain to stain of gram negative after removing our outer membrane.
After alcohol?
We understand?
Yes.
What is gram? Gram is
Stain.
the name of a scientist. Hans Gram. Christian Gram. But for gram staining in this section, the first step is smear preparation. What is smear, Fahad? What is smear? Do you read your lab text for section number 1 to bacterial staining?
Yeah, but it was long ago.
Take lab text from your friend.
It's the same. When we state.
Yes. Lab text and presentation, both. Each section contains 2 PDF. Presentations and lab text. This is.
We never had this.
You will have exam from this file. You understand?
Yes.
And next section you will have oral quiz. Ambrose. Yes.
Just this is
امروز یا فردا؟
The exam is
امروز and فردا.
اگه بشه برای فردا همش کجا بگیم؟
بعد از ظهرم کلاس داریم.
آره واسه فردا خوبتر نیست؟
فردا از
بعد
خوبه. بچه محلی دیگه هفته گوش میدن. تاجیکی یا افغانی؟
تاجیکی.
فارسی خوب صحبت. فارسی اصیل فارسی شما.
اون موقع همه جا بودیم دیگه الان جا
خب what is smear? Smear. What is this?
Smearing the
Smear.
Shake the bacteria.
Smear. Smear is a thin layer of bacteria on a slide.
blood.
Yes?
You smear.
To prepare a smear from solid media, such as this. Place a loop of water. Here, pay attention here. Sorry.
Bubble.
A loop of water on your slide. Okay? That is enough. Then, sterile your loop. And take very small amount of bacteria and mixed with water. Look at here, like this. Yes? Clear? Bale? This pattern. Yes. From center to around. On a stage.
So we use this for Edron?
Yes. This is a staging tray. Okay? Place your slide here. Don't place here. Here. After air dry.
تبي خيط عنكبوت
After air drying, the second step is heat fixation. Heat fixation. What is aim of heat fixation?
Fixing bacteria.
Yes. You.
break the
Look at here.
bacteria.
This S-like contain bacteria.
Yeah.
This bacteria can cause disease
Yeah.
and contaminate yourself and your friends. Huh? We must kill our bacteria before staining. Heating, we kill bacteria. 1. 2. We increase the attachment of bacteria on a slide. So, aim of fixation is killing of the bacteria and increase the attachment of bacteria on a slide. Okay? After air drying, for heat fixation, pay attention here, yes. Thank you. Yes. Move 3 times of your slide on top of the flame. Pay attention. Another. 1, 2, 3. That is enough. Most heating effect on your quality of your staining, and maybe change gram positive to gram negative. After fixation in gram staining, the first stain is crystal violet. Float crystal violet and bacterial smear. Time is 1 minute. Take your time, for example.
Okay.
1, 2, 3, 4, 6, 7 is 1 minute. After 1 minute, you must wash your slide. For washing, use water
Alcohol.
like this. Pay attention. Clear?
Yes.
Yes. Then, the second is iodine. Iodine. Add iodine.
Also on
Time 1 minute.
after the session.
Then is washing and number 3 is alcohol. The time of alcohol is very important.
Because it's one.
Use 5 to 7 seconds or 5 to 10 seconds. 1, 2, 3, 4, 5. Wash it. And final step is saffroning. Time of saffroning is 30 seconds to 1 minute. 30 seconds to 1 minute. Final step is washing. Look at here. Washing and drying by pour. You understand? Yes? Yes. Bismillah.
تا جای بریزیم
Don't use pour for smear drying. You understand? Bismillah.